In today's paper, we are going to look at supramodel role of basal ganglia in memory and motor inhibition. We all know that the basal ganglia is involved in motor actions, but the question remains if it plays the same role for memory, and this is what we are going to learn from this paper. A main region is said to perform a supramodel role if the abstract functionality is common across the different inputs. Here the two inputs that we are going to consider are the motor and the memory. The functions we are going to consider are prevention and cancellation. The process I'm going to take you in this paper is first we are going to ask the right questions, then we are going to find the right method, perform the experiments, do the analysis to conclude the results. So far we know that, and also as we have studied from our exam yesterday, we can recall that the basal ganglia is involved in termination and initiation of motor action through its indirect and direct pathway. A hyperdirect pathway from cortical to subthalamic nucleus exists that functions to prevent motor actions as well. Also, we have seen evidence that the PFC or prefrontal cortex perform a supramodel role inhibiting memory and motor. Now, what is the key research question that we are going to ask? Number one, is there a common mechanism in inhibiting motor and memory access? We are talking about the pathways from the cortex to striatum. And is the circuit the same? But when you ask this question, we can ask the two supplemental questions as well. Are the BG regions, the brain basal ganglia regions, common for the domains? Are there common regions for basal ganglia for the process? Setting up the right experiments to answer these questions is critical. So the author chooses three tasks that overlaps domains and process. The domains of the memory and the motor, and the process of prevention and cancellation. The first task, go no go, the user is presented with a visual stimuli and asked to perform a motor task such as pressing the button when they see green circle, and not to perform any task if they see a red circle. In the stop signal task, the users are again asked to perform the motor task irrespective of the color of the circle, but when they hear a audio cue such as beep, they need to stop the motor task. Thereby we are making this as motor cancellation. In the think no think task, the user is asked to memorize a set of cue targets. When the cue with specific color is presented, they are asked to remember the targets, otherwise do not recall the targets. Based on these three experiments or three tasks, we can devise three hypotheses actually. One is like if the supramodel mechanism exists, then these two regions should overlap. At least these two clusters should overlap. If the BG involvement is domain specific, then the first two tasks should overlap. If the BG involvement is process specific, then the second two tasks should overlap. Let's see what the method that they have involved. Here the author used meta-analysis, which refers to the statistical analysis that combines results from the previous studies. Note that the previous studies we need to find is basically that includes all the three tasks that we discussed earlier. From the imaging data performing the task, the author computed ALE. Just for the interest of time, we can imagine ALE as a common cluster of activity across subjects. Also, they perform the conjunction and conjoint analysis between the tasks, which we are going to show on the part of the results. Given the importance of the distinguishing the basal ganglia region across the different tasks, the author defined the region of interest manually to obtain more precision in the regions, which we are going to see how it helps in our results finding. The first is like here is the picture on the left side shows the ALE activation in the cortical region. We are going to analyze the results in three stages: cortical, subcortical, and domain specific. The picture shows the result of ALE indicating the activation across the subject in each of the tasks. First, we can see that the cortical region there is activity in all the three tasks. And also very interesting notice is that the DLPFC region of the cortex is activated in all the three tasks. When we do pairwise comparison, we see two conclusions actually. The number of regions that are active in the action prevention is not large, while when you have the action cancellation and the memory cancellation, the regions are quite extensive. And also one more key observation is like there is a significant overlap in the active regions in the cancellations task, and as mentioned earlier, the DLC is active across all the three tasks. Now next, when we observe the subcortical region, the first thing that we can see is like There is a reliable activation of basal ganglia in all the memory and action inhibition tasks. Memory and motor tasks, sorry. In the action cancellation task, the activity is clustered to the right hemisphere, whereas in the action prevention, the activity is clustered in the left hemisphere. I want to remind the question that we asked earlier, is the same basal ganglia region activating both action process? Now we know the answer. And thanks to manual region of interest separation in the basal ganglia, we now find that the total volume of activation in the BG regions for actual cancellation is much larger compared to the action prevention. Now when we compare the activities between the memory and motor cancellation tasks, several interesting observations arises. Both tasks show significant overlap in the BG regions. Although memory inhibition reliably activates several BG regions such as the caudate, anterior putamen, which are not active in the motor cancellation task. Again, here the key takeaway is that the memory inhibition activation overlaps significantly with the action cancellation rather than action prevention. To conclude, this meta-analysis is in line with the past analysis where the cortical region engages supramodel role for the inhibition task. Subcortical activations were quite interesting that even though the first two tasks were motor in nature, due to difference in process, they are activating different cortical regions. This is critical conclusion as they indicate different underlying cortical processes. As a grand conclusion, we can see that the BG is not only involved in motor inhibition but also in the memory inhibition. But as with any research, more questions get raised than answered. So some of the follow-up studies that can be done are in the memory inhibition, we saw STN, subthalamic nucleus were activated. So this needs to be further studied. We also observed that DLPFC is activated in all the three tasks. So is there a DLPFC striatal pathway? We need to do the further study on that. And the final message that we have is the basal ganglia are consistently activated in inhibition of both the action and thoughts. Its function is not limited to motor control but also in the higher cognitive process such as memory. Thank you for your time.